going to be day 2, and we're going to finish up this PowerPoint. So we'll go quickly through these notes. I believe this was the place that we left off. Just a little funny, selfie, sympathize, photosympathize, all that stuff. Now let's actually get to our notes. Up until this point, we've covered DNA, chromosomes, chromatids, centromere. That's kind of the structure of the DNA within the nucleus. Now let's actually talk about the different types of reproduction. The big thing to remember with asexual reproduction, besides the fact that it is only one parent, the big thing to remember is that it is identical. The organism will make a copy of itself to produce identical offspring. That means it's going to be a clone. It's going to be identical. It's just that you're copying the same DNA from one organism to the next, to the next, to the next. That is how asexual reproduction happens. There are several different types. We have binary fission, which is bacteria. Mitosis, that's what your body cells are doing. Most eukaryotic cells do this. Vegetative propagation is this picture right here. If you're looking, the vegetation, the plant, actually just drops off a piece of itself. And then budding is going to be what yeast does, and it kind of just, or hydra, if you're looking at this part right here, here's the hydra organism, and it's going to bud off a little piece of itself to become a new organism. So these are different types of asexual reproduction that we're going to cover a little bit, but the main thing is it's always identical. There is no variation whatsoever in this. All right, binary fission. This is bacteria. This is prokaryotes. This is if you remember, pro means no, which means they have no nucleus. Instead, they have this nucleoid region, which has the DNA just kind of out there, and then it will copy itself. The cell will continue to grow larger and larger until it's made that copy, and then by doing that, it'll split in two right here. And when it splits into two, that is binary fission. It goes from the one parent organism to having the daughter cells then. So from one bacteria, that becomes two. From those 2 bacteria, you'll get 4. And from 4 bacteria, you'll get 8. And it kind of multiplies and multiplies. But the main thing, again, identical. No change whatsoever. It's just the cell is getting larger and then it splits itself. The DNA is going to be exactly the same. For vegetative propagation, it's another form of asexual reproduction, but mainly in plants. Vegetative, vegetation kind of deal. Part of the plant detaches from the parent plant to form their own organism. But if it simply detaches from that organism, it's still carrying the same DNA. So it's still going to be genetically identical to the parent. Like I said before, budding, this is also asexual reproduction. It's just a small piece, starts growing from the parent until it gets large enough to break off and become its own organism. But it's still same DNA, it's growing and growing until it splits off. So you have genetically identical organisms. Sexual reproduction, that is going to have 2 parents. That is what we are. That is what animals are. Plants are. You're having a mom and a dad who are giving 2 separate bits of genetic information to form a whole. So where we have now 2 for sexual reproduction, the big thing is it is genetically diverse, which means that's going to have some changes in there. There's going to be a difference between it. So we are not going to be exactly like our mom, and we're not going to be exactly like our dad because we're getting a mix of the 2, and that is sexual reproduction. This happens in the sex cells, the sperm and the egg, which are called gametes. So our sperm and egg are called gametes. And that is going to be the difference. We have sex cells, which are gametes, and body cells, which are somatic cells, if you recall. So we're actually getting into the cell cycle now. We've talked about the DNA and the chromosomes and reproduction, but how is the cell actually dividing? What is it going through? What is the cycle? And this is what it's going to be talking about. We have interphase, which covers G1, S, G2, and then we have mitosis, which is prophase, metaphase, anaphase, and telophase. What I like to say is PMAT. And you can actually, this is a little bit wrong because we also need to have our cytokinesis in here, which is where the cell itself, the cytoplasm, is going to split. But this is just kind of an overview, this diagram, of what is happening in the cell cycle. All right, the cell cycle includes three main parts. You have interphase, mitosis, and cytokinesis. So mitosis is the nuclear division, or where all the DNA is held inside the nucleus. Mitosis is going to split that in half. That's where it divides. And then cytokinesis is the division or the dividing of the cytoplasm, which is the rest of the cell. All right, let's start with the first one, G1, S, G2. This is the main part of the cell cycle. It's just it's the longest stage of the cell cycle. And we say it's resting because the cell isn't going to be actively dividing here. Instead, it's going to be growing. The DNA is going to be replicating. Cell organelles are being made, and it's going to prepare itself for the cell division come from mitosis and cytokinesis. So right now, during interphase, the cell is just kind of chilling out, doing its thing like you do. You're all simply eating and growing and just doing whatever you do. And that's what the cell is doing. So inside interphase, you have G1. And the big thing, G1, is all about growth. Okay? So G1, you have the main growth stage. The cell itself is going to be increasing in size. And because it's going to increase in size, it means it has to have more organelles. So it's got to have more organelles made. And then S phase is DNA replication, and this is synthesis. Okay, so it's copying all these instructions through DNA replication, and where it's in the form of chromatin, if you remember, that is like the spaghetti, but we're just replicating or making copies of that DNA instruction so we can pass it on. And then G2 is where it's going to prepare. That's what we remember about G2. It is preparing for mitosis. It grows a little bit and it makes more proteins because the nucleolus is about to disappear. But the main thing is it is preparing for mitosis. And now let's get into mitosis. The division of nucleus occurs first in mitosis. So mitosis is actually asexual reproduction, meaning it's still one parent cell. That's what we call it. We have a parent cell, and that parent cell will divide until it becomes two daughter cells, but they're simply copying and splitting the DNA, so it's going to be identical. Daughter cells are genetically identical. That's why it's called asexual. Mitosis consists of 4 different stages: PMAT, prophase, metaphase, anaphase, and telophase. So those are the ones that we really need to remember. 4 different stages: PMAT, pro, meta, ana, telophase. Here's a diagram of what's really going on. If we start here at the very beginning, we have prophase, where our centrioles are beginning to appear. They move to the opposite sides of the cell. The nuclear membrane is going to disappear. The nucleolus disappears. And we see the DNA is now in the shape of an X for chromosomes. Then we're going to metaphase. Meta, middle, M, middle. So your metaphase is where all the chromosomes are going to line up in the middle of the cell. And then we go to ana, and ana apart, A. It's kind of how you can remember it. But this is where those chromosomes are going to be pulled apart from that middle section, and where they are duplicated with sister chromatids. Now you're getting the separate ones, but they are now called chromosomes. That's anaphase. And then we're going on to the last part of mitosis, which is telophase, is tele2, TT. Okay, so tele2 gives us 2 nuclei. So that's important. It starts elongating, getting ready to divide through cytokinesis. And we're now getting their nuclear membrane to reappear. The nucleolus reappears. And the DNA is going to become chromatin now. So what are the stages? Let's go over this in a little more detail. We got pro, meta, ana, telophase. Prophase, the longest phase of mitosis. Not that that's saying a whole lot, because interphase takes the longest throughout the cell cycle. But for mitosis, which is the nuclear division, prophase is the longest. Here we have our nucleolus is going to disappear. That chromatin, the spaghetti that we were saying, the DNA is shaped like spaghetti, starts to condense into those chromosomes consisting of 2 sister chromatids, making the X for us. Our centrioles, which are, they look like licorice in the diagrams, they're going to move to opposite sides or to move apart in the animal cell, not found in plants. And then the spindle fibers are going to kind of come away from those centrioles so that they can attach to the center part of those chromosomes, which are called the centromeres. And where it's attaching at the centromeres, it will later be able to kind of pull them apart. So it's getting ready for the next few stages. And of course, the nuclear membrane disappears so that it can later become 2 separate nuclei. So we do have 2 different stages. There's an early prophase and a late prophase. And if you can kind of see, you have your centrioles off to the side here. Here's our centrioles, here's your centrioles. They're going to move in from early to late so that they're going to the opposite sides. In early prophase, this is your cell membrane, nuclear membrane that's going to start disappearing. This nucleolus is going to disappear as well. And that's what we see here happening in our late prophase. So this is the only one that is going to be split up like that. All right, here's an example. If you can really just pay attention, go back to your notes. The Canvas is on PowerPoint. Look at those images. All right, metaphase. This is going to be the shortest stage simply because all they're doing is lining up in the middle of the cell. So the chromosomes line up across the middle of the cell, the equator, center, and the centromere of each chromosome attaches to a spindle fiber. So the spindle fibers kind of push them and align them in the middle to get ready to be able to pull them apart. So meta means middle. They're lining up in the middle. There's your image of the cell. Now anaphase. These sister chromatids are coming together in our chromosomes to make that X shape. Well, they're going to be pulled apart, and then after separation, the chromatids are now considered chromosomes. So during this phase, the cell contains twice the normal number of chromosomes because it's been duplicated or replicated beforehand, and now's the time where it's getting ready to split, so there are twice as many chromosomes as there are usually. The cell begins to elongate. That really just means that it kind of stretches out to get ready to divide. At the end, there are an equal number of chromosomes at these poles. And again, the poles are the opposite ends of the cell. Here we go again. Anaphase, they are no longer sister chromatids, but they're chromosomes. They're being pulled apart or away from that middle line. There's an image. And telophase, tele means two. So the nuclear membrane reappears around the two nuclei. The nucleolus reforms in each of the nuclei, and the chromosomes uncoil and appear as chromatin. In the end, we have two genetically identical nuclei, and then the next step is cytokinesis. So if you kind of are paying attention and looking back to it, prophase and telophase are exactly opposites of each other. Prophase, your nuclear membrane disappears, nucleolus disappears, the chromatin coils up, condenses to become chromosomes, and for telophase, that membrane reappears, the nucleolus reforms, and the chromosomes uncoil or spread out to become chromatin. So they are the beginning to get it all started, and the opposite, telophase, to end it. Here we go. This is another diagram for it. And then cytokinesis. Cytokinesis is going to be the final stage of cell cycle, because where we have now two nuclei within the one cell, we need to divide the cytoplasm and the organelles within that cell to make the two daughter cells. So cytokinesis is the division of cytoplasm of the cell and its organelles into two new daughter cells. Now there's going to be a difference in how cytokinesis happens within a plant and an animal. An animal is going to have a cleavage furrow, which really means that it seems like there's a, imagine a string wrapped around a cell. And that string is going to pull tighter and tighter and tighter until it just cleaves or cuts the cytoplasm in two. Now that is how the cell membrane is going to form to make those two daughter cells. Now the plant cells, they have that cell wall which allows them to maintain their shape and give them protection. Well, that's going to be a big problem. There's no way that they can have a cleavage furrow where it just kind of pinches. Instead, they have a cell plate. And that's pretty much where a new cell wall is going to form between the two daughter cells. So here it is again. The animal cell has that cleavage furrow, which is where the cell membrane just pinches until it completely makes the two cells. And the plant cell will create that cell plate or reform a new cell wall. So how do we have this starting out? We have our parent cell that will go through interphase. And then our chromosomes are going to be copied and doubled in number. Then we're going to have prophase to get them all ready to divide. Metaphase gets them in the middle. Anaphase pulls them apart. And telophase gives us two nuclei with cytokinesis, making it two new cells. I'll let you go through this on your notes on your own. But this is important. Cancer is really uncontrolled mitosis. Mitosis must be controlled, and otherwise growth will occur without limit, which is how tumors form. Control is by a special protein produced by oncogenes. So that is just some random fact there, that if mitosis isn't controlled, it does lead to overgrowth and over-reproduction. So it does lead to tumors and cancer. So go through the rest of this Canvas slide and file on your own to go through the different stages again. But I hope this helps and gets you ready for your quiz. Thanks.